Dear students, welcome to Tungal online classes. Today I am going to start with a new cell organelle, that is centrosome and centrioles. Right? So what is this centrosome? So what are these centrioles? The difference, it confuses us sometimes. Right? So first of all, we will go through the centrosome entirely and later centrioles. Centrosome is nothing but it is densely stained or distinctly stained spherical cytoplasmic area that contains a pair of cylindrical centrioles. Right? It's called as centrosome. Very clearly we can write. So what is centrosome? It is a densely stained or distinctly stained cytoplasmic area which contains a pair of cylindrical structures called centrioles. What is centrosome and what is centriole? The difference you come to know in the definition itself. Means here the centrosome is the cytoplasmic area which is a densely or darkly stained area in which a pair of cylindrical structures called as centrioles are lying. Right? So that is centrosome. Okay. So now it is very clear to you people. Right? So let us see occurrence of these centrioles. Of the centrioles. In what type of cells centrioles are usually present? Let us see now. So centrioles are usually present in the animal cells. So they are usually present in the animal cells. They are absent in angiosperms, especially angiosperms, and they are also present in so lower plants. Okay, so this is more important aspect. This makes the difference between the plant cell and animal cell. The presence of centriole, so in animal cell, and absence of plant cell, this makes the difference between plant cell as well as animal cell. Right? So one. Now, what is the area that contains a pair of centrioles? Already you know, densely stained area which is a little bit fibrous, which is somewhat distinct from the other cytoplasmic areas itself, and that kind of area which is lying very nearer to the nucleus. In diagram we can observe that. See here, this is one animal cell and this is the nucleus. Near the nucleus there is a cytoplasmic area which is a densely stained area, and in that area what are lying? A pair of cylindrical structures called as centrioles are lying, and that densely stained area itself is called as centrosphere. What it is called as? Centrosphere. It is darkly or densely stained area near the nucleus. Darkly stained area near the nucleus. In that area there lies a pair of centrioles. So I hope it is very clear to you now. What is that area called? Centrosphere. Now centrosphere and centrioles together form centrosome. Together form centrosome. See here, centrioles plus centrosphere together form centrosome. What it is called as? Centrosome. Okay, now it is very clear. So how is the structure of each centriole? There is a need of study of the centrioles. How they are lying, how is the structure of the centrioles? Let us discuss now. See here is one centriole which contains 9 triplet structures. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplet tubules it contains. See here. Right? Okay. Such kind of structure we can observe here. So it is a cylindrical structure which is lying right angles to another centriole. This is one centriole what I am showing now. Right? So which contains 9 cylindrical triplets. 9 cylindrical triplets which are peripherally arranged one. See here, these are 9 in number, they are peripherally arranged, and so they are triplet in number. I am not showing here triplets, but I am going to show here the cylindrical structure over here. And such kind of a pair of centrioles are lying in the centrosphere forming the centrosome. Is it very clear? So let us see the same diagram will enhance over here now. So the same diagram will go to enhance over here. So how these 9 triplets are arranged over here in the structure, let us see. So this is one cylindrical structure that contains how many triplets? 9 triplets. See here, this is 1, this is 2, this is 3, this is 4, 5, 6, 7, 8, 9. 9 triplets I am going to show you here. See here, it has 1 and 2, 1 and 2, 1 and 2, 1 and 2. Means totally here you are going to see 9 triplet microtubules you are going to see over here. See this kind of 9 triplets you are going to see over here. They are peripherally arranged one. They are peripherally arranged one. So these 9 triplets are just like this. See here, this is one triplet. Sorry, this is one triplet. What I am going to draw over here. This is one triplet. Means 3 cylinders are present over here. So 3 cylindrical shaped microtubules are lying just like this, and they are named as A, B, and C. Okay. So see here, this is one triplet. Such kind of 9 triplets are peripherally arranged over here, right? And this A is made up of 13 protofilaments. What is this A made up of? 13 number of protofilaments. They are also cylindrical proteinaceous structures. Microtubules are the tubular cylindrical structures which are made up of a protein called as tubulin. Right? See here, it is made up of 13 number of protofilaments, and they are helically arranged over here. A 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So 13 number of protofilaments is made up of, and these two are made up of respectively 10 protofilaments. How many protofilaments? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10. And this is also say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 number of protofilaments they are respectively made up of B and C. But A is made up of 13 number of protofilaments. And respectively, these are interconnected. The protofilaments are interconnected. So there are 3 to 4, I mean 2 to 3 number of, so the protofilaments are shared between these triplets. Right? This one 3 to 4 which are adjacent to B, they are sharing with B. And from B, C, they are sharing with C. Like this, 3 triplets are arranged over here. They are made up of protofilaments, and so these 3 are made up of the protein called as tubulin. Why? Because these are called as microtubules. Remember. Okay. So here we consider this is A, this is B, this is C. In all these are respective A, B, C, A, B, C. And one thing we should remember here, A is connected to C. A is connected to C of adjacent triplet. Right? See, this is A, this is connected with the C. This is A, this is connected with the C. And this is A connected with the C. This is A connected with C. And this is A connected with C. This is A connected with C. A connected with C. And A connected with C. See here how it looks like. It looks like a carved wheel. It is looking like carved wheel, right? So this kind of arrangement is seen in one what? Centriole. In one centriole, this kind of arrangement is seen over here. And all these peripheral triplets are interconnected with the centrally located proteinaceous structure, and that centrally located proteinaceous structure is called as hub. Hub it is called as. To this hub, so all these are interconnected with the help of a thickening which is lying to the A. This is A. Nearer to A there is a thickening. This is also A. Nearer to that there is a thickening. And here is also thickening. Here is also thickening. A thickened body is lying near the A. See here. Lying near the A. Right? So all these are connected. Means these are named as X bodies. What these are known as X, or they are called as foot. They are called as foot or X thickening. They are called as, and these are connected to the centrally located hub with the help of a radial spokes. See here, this is radial spoke, radial spoke, radial spoke, radial also radial spokes. With the help of the radial spokes, all the eggs are connected over here. See here, this thickening X is connected with the AC linker. What is this? This is C. This is called as AC linker. What is that? AC linker it is called as. The structure which connects A to the C, it is called as AC linker. In between the two X bodies, there is another thickening that we come across. See here. This is another thickening. And this line between two X bodies are, so two A's over here, two X bodies in between, there is a thickening, and this thickening is called as Y thickening. So it is lying between two X, right? This is also another one, this is another one, this is another one. So like this, Y thickening is also seen over here. Observing that, so there is another thickening which is lying between two X bodies or X thickenings, and that is called as what? Y thickening. That is called as Y thickening. See here, these all A are interconnected with the Y. See here, this is connected with Y. This is connected with Y. All A are connected with the Y. See, all A are connected with the Y. And this Y is connected with the AC linker. This Y is connected with the AC linker. See here, Y is connected with the AC linker. See here, Y is connected with the AC linker. Okay, so this is how the structure of each centriole is. So imagine this is the cross section we will have to imagine. So the same structure which forms the cylindrical structure. Such kind of, so 9 triplets what we come across, they are all elongated one, they are cylindrical one, they are along the central axis itself, right? This kind of structure you can observe in each centriole, right? So each triplet is made up of a protein called as tubulin, and each one is called as microtubule. Each one is called as microtubule. Such kind of 3 microtubules. So together form what? So triplet. One triplet like this. So this is how the structure of one centriole is there. Such kind of a pair of centrioles are lying in the centrosphere, forming the centrosome. Right? So I hope it is very clear about what? The structure of one centriole, right? So this is one centriole, and what are the major functions of these centrioles? Let us see now. The major function of these centrioles is nothing but they act as the basal bodies, right? Basal bodies of what? Basal bodies, they give rise to cilia as well as flagella, right? These form basal bodies which give rise to cilia and flagella. This is one function of the centriole, and another major function of organization of what? During cell division, you know. So there the chromosomes are hanged to spindle fibers. Those spindle fibers are usually formed by the microtubules, which are formed from this centrioles, right? These centrioles help in organizing the spindle fibers during cell division. That you can observe over here. If this is the animal cell, so here is the centriole, here is also centriole that organizes what? The spindle fibers like this, to which the chromosomes are usually held over here, right? Chromosomes are usually held over here. See here. Chromosomes are held over here. This is the centromere, which hangs to the spindle fiber. This is spindle fiber, right? So this is how spindle fibers are formed from these centrioles itself during cell division. These are the functions of these centrioles. And next one is very important one, cilia as well as flagella. So how these are structurally and what are the functions? So they are nothing but locomotory organs in lower organisms. So where they are actually occurring. So all these are the things let us discuss now itself. Next one is cilia and flagella. Cilia and flagella. What these are, you know it better, right? Cilia as well as flagella, these are nothing but they are the fine hair-like vibratile, right? So filamentous structures which act as the locomotory organs in lower organisms, as well as they also help in movement of the substances in one direction, right? So such kind of structures are protoplasmic processes itself, right? And such kind of structures are called as cilia as well as flagella. Let us write the definition for this one. These are fine vibratile protoplasmic processes that or which help in locomotion of lower organisms and also help in movement of substances in one direction. And such kind of protoplasmic vibratile processes called as what? They are cilia as well as flagella, and these create a kind of current in the water, so that help the animal to move in one direction, right? So these move and create a current water especially or in a liquid medium, any liquid medium. Why? Because bacteria usually they are grown in one petri plate. So in one medium itself, that medium may not be water, so it is one kind of medium, sugar medium or something else, right? That's why. So in water or any other liquid medium, so which help in movement of body of organisms, right? So these are nothing but cilia as well as flagella. So cilia are smaller in size when compared to flagella. They are smaller in size and they are more in number. More in number. But flagella are longer in size. They are lesser than the cilia itself, right? So this is the one more difference between these two, right? And the size of the cilia itself is smaller. The size of the flagella itself is little longer than the cilia itself, right? So next one. So occurrence of cilia, first of all we'll write. Okay, so where do you find these cilia? So already you know in the protozoans ciliata is one of the groups that we come across. So in the ciliata group of protozoans, cilia are the locomotory organs in these organisms. One is ciliata, and the next one is the flatworms, which have the flame cells of flatworms. Flatworms also have these cilia. And the next one, the larvae of some of the lower organisms, that is invertebrates. Larvae of invertebrates also have these cilia as the locomotory organs, and these are also present in some of the animals, higher animals. So in some tissues, especially epithelial tissue, ciliated epithelium, you know it better, the epithelium of respiratory tract, right? Respiratory tract, you know which one are present? Cilia are present. So these cilia are also present in the respiratory tract as well as renal tubules. They are also present in renal tubules along with oviduct. They are present in oviduct also. So these being in oviduct, renal tubules, the respiratory tract, they help in movement of the substances in one direction. Being in these one, being in epithelial tissue, so these cilia would help the movement of substances in one direction. Already I have shown you the oviducts in which the egg cell is moving, right? In what direction it is moving? So it depends on the cilia present there, right? So they are the cytoplasmic processes, so which are formed at the free surface of the cell in tissues, right? So all these are the things I have told you whenever we were dealing with the tissues, animal tissues, that's all. So occurrence, this is about the cilia. What about the flagella? Where do you find these flagella? So flagellata is the group of the protozoans. Flagellata is the group of the protozoans. In these protozoans also you will find what? These fine filamentous protoplasmic extensions. They are nothing but flagellas. You can find flagellata group that includes what? Euglena is the best example for this one. Okay, so flagellas are present there. And choanocytes of the sponges. Sponges, you know, in sponges choanocytes have flagella. Alright? Choanocytes, apart from this one, gastrodermal cells of cylinders. Gastrodermal cells of Cylindrita. So there also you will find what? The flagella. Along with this one, human sperm, you know. Sperm has the flagellator structure, right? Tail itself is just like a flagella, so it helps in movement of the sperm, right? There also you will find, and it is also found in the male gametes of some of the lower plants like where algae and bryophytes as well as pteridophytes. You will find these what? Flagellae. And apart from this one, zoospores. Right? In zoospores also you will find these protoplasmic extensions, that is flagella. Right? So this is about the occurrence of these flagellas. So almost structurally these cilia as well as flagella are similar in their structure. Right? So what are those structures that we come across in the structure of cilia as well as flagella? Let us see now. Structurally, these cilia as well as flagella has three major parts. One is basal body, second one is basal plate, and third one is shaft. Are the three major parts of this? So, what? Cilia and the flagella, right? Structure of cilia and flagella. Almost both are similar in their structure. Both are similar in their structure. Okay, so the structure of cilia and flagella, structure has three major parts. One is basal body and basal plate, and third one is shaft. So like this there are three parts. One is basal body. Let us see what is this basal body. As you know, already we have studied the centrioles. Centrioles are responsible for the formation of the basal body for cilia as well as flagella. Means the basal body is derived from what? The centrioles, right? So basal body, it is the structure derived from the centrioles, right? Centrioles, which is lying in the cytoplasm, right? So this basal body is lying in the cytoplasm below the plasma membrane. There it is. Okay, let's see what is basal plate. Basal plate. It is the structure structure that we have to compare with the centriole as well as the cilia as well as flagella. So this is more important as basal plate. So it is at the level of plasma membrane. It is at the level of plasma membrane. It is at the level of plasma membrane, and this so is having the doublets in the sense that 2 microtubules. 2 microtubules it is made of. Doublet, it means doublet. So already we have seen triplets as A, B, C. 3 microtubules together form the triplets, which are arranged peripherally, right? So that is the thing what you should remember here. Here instead of triplets, only doublets will find over here. So this is B and this is A. So these two together form what? The doublet. These two together form what? Doublet. It forms the doublet structure. Here the third one, that is the C microtubule, is so related. Means here microtubule that appears, that disappears over here. Microtubule C that disappears over here. Only doublet is seen over here instead of triplet. And so 2 new singlets are formed at the central axis. Right? 2 new singlets are formed at the central axis. They are attached with the central sheath. So how they are attached? So in what way the structure is? Let us see in the entire structure of cilia as well as flagella. So now we came to know there are 2 parts. One is basal body and another one is basal plate, in which only the doublets are there. They are also 9 in number and they are also peripherally arranged over here, and here the third microtubule, that is microtubule C, disappears over here. Instead of that one, centrally located the 2 singlets are formed over here. So this is basal plate, so which is at the level of the plasma membrane, which is lying at the level of plasma membrane. So next to the plasma membrane, outside the plasma membrane, there starts elongated cylindrical structure that forms the shaft. Right? What is this shaft consisting of? So let us see. So theoretically first and later diagrammatically. Okay. So shaft has the surrounding membrane. The surrounding membrane called as plasma lemma. The surrounding membrane called as plasma lemma. One thing. And the next one. What is the next one? Plasma lemma is over. So means peripherally what is there? There is a membrane around the cilia as well as flagella. Imagine yourself, this is one bacterial cell which has a pair of what? So flagella are there, and this is the plasma membrane. Imagine below the plasma membrane what are present? So basal bodies are there, and the next one is the basal plate, followed by this is the shaft. What it is for us? Shaft it is for us. This shaft, so when you will find this structure larger, so that moment you will find this bit of structure. So this is, when you take the cross section, it looks like this. This is nothing but plasma membrane. Outermost plasma membrane is there, and inner to this plasma membrane. There is a matrix within it. There is a matrix within it. Matrix is present over there. For the nourishment, matrix is present. And along with this one, there we come across another structure which consisting of 9 pairs of doublets. 9 doublets like this, and all these are interlinked over here, and centrally located 2 singlets are also there, and all these are interconnected with the doublets over here, right? So this is nothing but axonium. This is nothing but axonium. This way there are 3 major parts are seen in the shaft. One is plasma lemma, and second one is matrix, and the third one is axonium. Axonium. All these are the things that we see diagrammatically first, and later we'll explain all those structures also. Now the structure of cilia and flagella diagrammatically. See here, the outermost single membrane will come across, and this is called as what? Plasma lemma. Outermost membrane is called as plasma lemma. Inner to this one, there you come across the cytoplasmic structure, that is nothing but matrix you will come across. Is the second structure. There inside that one, 9 doublets. See here, this is 1, this is 2, this is 3, this is 4, 5, 6, this is 1, this is 2, this is 3, this is 4. This is 5, this is 6, only 6 are there. Yes. This is plasma lemma. This is plasma lemma. Inside this one there is a matrix. There is matrix, and we will come across 9 doublets. 1, 2, 3, 4, 5, 6, 7, 8, 9. See here, 9 doublets will draw here. This is one doublet. This is another doublet. This is third doublet. This is fourth doublet. This is fifth doublet. This is sixth doublet. This is seventh one. This is eighth one. This is 9. So like this, 9 doublets that you come across over here. And these doublets are interconnected with the 2 singlets at the center. 2 singlets at the center. This is one singlet, this is another singlet. And this system itself is said to be as 9 plus 2 system. But in case of the centriole, there you come across 9 plus 0 system you will find in centrioles. In centrioles 9 plus 0 system, but in case of celiac flagella 9 plus 2 system you will come across. So these two are interconnected with the sheath, and that sheath is called as central sheath. It is called as central sheath. It is called as. To this singlets, all these doublets are interconnected. But one thing we should know here, what is that is, So doublet has one is A and the other one is B, right? So doublet microtubules these are. So whenever you see doublet, this is A, this is B. B is a little wider than the A, and A is made up of 13 number of protofilaments. How many? 13 number of protofilaments, and this is made up of only 10 number of protofilaments. So this is B, this is A, right? Point to be noted over here, right? Such kind of two doublets, 9 pairs of doublets that we come across, and all these 9 pairs of doublets are interconnected with the singlets which are located at the center with the help of radial spokes. With the help of radial spokes, you are observing here radial spokes. So that connect these doublets to the central axis. See here. So these are called as radial spokes. These are called as radial spokes. They are called as. Okay. So this is nothing but the central sheath, and this is nothing but singlets, and this one is nothing but doublet. See here, so there is a link between two doublets, right? There is a link between two doublets. Inter-doublet proteins these are. Inter-doublet linkers these are. Inter-doublet linkers these are. And so we have to observe here, A has two extensions of the protein, and these extensions, one of them has hook-like structure. It has hook-like structure. Every A has a hook-like structure and a normal one. A has a hook-like structure and normal one. So A has a hook-like structure and normal one. Like this, there are two extensions that we come across. During the movement of this cilia as well as plasma, so this hook that touches the B of the adjacent doublet, and when it links that movement, it helps in movement. It attaches to this one and helps in movement itself. So that is the use of this hook-like structure. Okay, so this kind of structure we will come across in the cilia as well as flagella. Okay, so these are the structures that we come across, and some more structures like very interesting one. Let us see here. So there already I have told you there are some two extensions that comes from A, right? And these are among these one, A has one hook-like structure which is outer one. This is inner one. Consider inner one. So and these are said as arms. What these are called as arms. A pair of arms that extend from A. So microtubule A here. And these arms as well as the central fibrils. These central fibrils as well as arms both are made up of a protein called as dynein. Both are made up of protein called as dynein with one enzyme called as adipase. Right? There is a need of energy for the movement of these flagella as well enzyme won't help in formation of the ATPs itself, right? That's fine. And one more thing, these are the radial spokes what we come across here. These radial spokes and interdoublet linkers, or these are also called as BA. This is B, this is A. Linkers, these are called as BA linkers. Linkers, these are also called as. These BA linkers as well as the radial spokes are said to be are made up of a protein, Naxin protein that forms these interdoublet linkers as well as the radial spokes. And one more thing we shall explain here. So regarding this radial spoke, whenever it comes towards the axis, it becomes broader. It becomes broader. That's why this part of the linker itself, the radial spoke itself, is called as the head part. Alright? So these are other things related with what? So related with this axoneme. This entire part forms the axoneme, and this is the matrix, and this one is plasma lemma. These are the three parts that we come across in this flagella as well as cilia, right? So what are these cilia as well as flagella help for? So let us see the functions of these one respectively now itself. See the functions of this flagella as well as cilia here. So first one. So already we know these are acting as the locomotory organs in some of the lower organisms. These act as locomotory organs in some lower organisms, especially animals. Example for this one is protozoa. Right? Protozoans are the organisms which use cilia as well as flagella for their movement. And second one, so in sperms, it helps in movement. Right? It helps in movement being in sperm. Next one. In some of the organisms, these cilia as well as flagella, they help in collection of the food material by creating a kind of water current inside the water. These create a water current to collect food and even to remove the waste materials from the body. Example for this one is some of the sponges, cylinderates, right, tunicates, right? All these are the examples for this one. They help in creating the current within the water, and through that current the food comes towards it, and the capturing of the food is done by the animal, right? So fourth one, being in the oviduct, it helps in movement of the materials or the ova in one direction. Being in oviduct, this help in movement of ova or egg cell, right? This is one more fact. Okay, so these cilia, what are present in the respiratory tract. So there they remove, they prevent the entry of the solid food or solid materials, right? So these help in removal of solids in the respiratory tract. Right? They trap those solid materials, maybe dust particles or some other microbes. So they have been trapped. So in the respiratory tract. So these are some of the important functions of these cilia as well as flagella. By this we finished. One is centrosome and the one is called as cilia as well as flagella. So these are the two structures that we finished, and in the next video we will start with the very important cell organelle, that is master cell organelle nucleus, in the next video. Thank you.